and regeneration declines. A primary reason for this is the depletion and dysfunction of our adult stem cell populations. Stem cells are the body's master cells. They're undifferentiated cells that have the remarkable potential to develop into many different cell types in the body during early life and growth. In adults, they act as an internal repair system, dividing essentially without limit to replenish other cells as long as the person is still alive. For example, hematopoietic stem cells in our bone marrow continuously generate all the cells of our blood and immune system. With age, however, the number of our stem cells decreases and their functional capacity diminishes. They become less efficient at dividing, differentiating, and responding to injury signals. This decline in stem cell function is a major contributor to age-related frailty, poor wound healing, immune senescence, and the degeneration of tissues like cartilage in our joints or neurons in our brain. The logical therapeutic approach, therefore, is to try and restore a more youthful and functional stem cell pool. Stem cell therapies for anti-aging generally fall into two categories. The first involves harvesting stem cells, often mesenchymal stem cells from sources like bone marrow or fat tissue, expanding them in a lab, and then infusing them back into the body intravenously. The theory is that these infused cells can home in on sites of inflammation and injury, where they can promote repair. Interestingly, much of their benefit may not come from differentiating into new tissue, but from their powerful paracrine effects. They release a host of growth factors and anti-inflammatory molecules that create a regenerative environment and wake up the local resident stem cells. The second, more advanced approach involves regenerating or rejuvenating our endogenous stem cells within the body. This could involve using drugs or growth factors to stimulate dormant stem cell pools to become more active, or using gene therapy to correct age-related defects in the stem cells themselves. This is a less invasive and potentially more targeted approach than systemic infusions. For instance, research is exploring ways to specifically rejuvenate the hematopoietic stem cells to restore a youthful immune system, a concept known as immune rejuvenation. While the promise of stem cell therapy is immense, the field is currently plagued by a great deal of hype and unregulated clinics offering unproven treatments. It's absolutely critical to distinguish between rigorous FDA-approved clinical trials and the services offered by these clinics, which can be both ineffective and dangerous. Legitimate research is ongoing for specific conditions like osteoarthritis, heart failure, and neurodegenerative diseases, with some early trials showing potential. However, the use of stem cells as a general anti-aging treatment is still highly experimental and not yet supported by robust clinical evidence. One of the most provocative and almost vampiric concepts in aging research stems from a series of fascinating experiments known as heterochronic parabiosis. In these experiments, an old mouse and a young mouse are surgically joined so that they share a common circulatory system. The results are striking. The old mouse begins to show signs of rejuvenation. Its muscle and liver tissue begin to repair more efficiently, new neurons form in its brain, and its cognitive function improves. Conversely, the young mouse exposed to the old blood shows signs of accelerated aging. These experiments strongly suggest that there are factors circulating in the blood that can either promote youthfulness or drive aging. The blood of young animals appears to contain pro-youthful factors that can rejuvenate old tissues, while the blood of old animals contains pro-aging factors that can impair the function of young tissues. This has kicked off an intense scientific race to identify exactly what these factors are. Is it the absence of old factors or the presence of young factors that matters more? The evidence seems to suggest it's a bit of both. This has led to the exploration of plasma-based therapies in humans. Plasma is the liquid component of blood that contains all these circulating proteins, hormones, and signaling molecules. One therapeutic approach is plasmapheresis, or plasma exchange. In this procedure, a patient's blood is drawn, the plasma is separated and discarded, and the red and white blood cells are returned to the body along with a replacement fluid, such as albumin. The idea is to dilute or remove the accumulated pro-aging factors from the old plasma, thereby reducing their negative influence on the body's tissues. Early clinical trials investigating plasma exchange for conditions like Alzheimer's disease have shown some tantalizing but preliminary results, suggesting it might slow cognitive decline. The research is still in its infancy, but the concept of cleaning the blood of deleterious aging factors is a compelling one. Another approach, which has garnered more controversy, is the direct transfusion of plasma from young donors to older recipients. While a few small-scale commercial ventures have offered this, there's currently no robust clinical evidence to support its efficacy, and major regulatory bodies have warned against it. The future of this field likely lies not in wholesale blood exchange,